All right. Hello, everybody. Hope you guys are having a good day and aren't too stressed from our exam yesterday. Um, if you've noticed, I guess I don't remember if I sent out a message or not, but I was able to upload the grades from the lecture exam that you guys took yesterday. I was able to get through all of them during my nutrition exam today. So in the gradebook, you should see a column for the Unit 1 lecture exam grade. The points that are showing up there do not contribute to your total points yet. This percentage out of the 50 points that were possible on this lecture exam that was converted to a percentage based on your score, that percentage and the percentage from your lab exam will average to become your score for the Unit 1 exam. And that is what will contribute to your total points out of 100. So yeah, that Unit 1 lecture exam grade is posted for you. Did some quick analysis of it. So with everyone who took it, the average was 65%. If we take out the two lowest grades, which were almost significant outliers, the average is around a 73%. So honestly, really good, and I'm really happy with that. Not completely happy with some of my questions and how they were worded, and I could tell that the goal with the question didn't get across with how some of the answers were. But overall, really, really well. The highest score was a 94, I believe. Yeah, a 94. So great job. I want to give us the opportunity to review those questions and talk about them at some point. And we may get to have some time at the end of today's lecture, and that could be something we use the rest of that time with. But the goal for today, or the main plan for today, is to get through chapter 5, which is all about the integumentary system, aka our skin. And we'll be talking about more than skin in this chapter, in this PowerPoint. The skin is technically just these top few layers here, what we consider our cutaneous membrane. So the epidermis and then the two layers of the dermis, that is what actually makes up the skin. But we'll be talking about all of the structures that perpetuate throughout those three layers, and then a bit about the hypodermis as well, which is technically part of the system, but again, not considered the skin. Right? It's basically a layer of fat, a layer of adipose tissue, some connective tissue there. So there's a lot of specifics in this chapter, of course, a lot of structures to learn. And like we did with chapter 4, I don't intend to spend time just reading all the names or reading just info that's already available and in front of you, and want to spend as much time as possible talking about the concepts, the functions, the physiology part, how it actually connects to that. So yeah, that's the plan for today. Do you guys have any questions or concerns before we get started with this content? Cool. Of course, there will be time after to talk about any questions as well. So I'm going to keep it in this view. I do have some info in the notes portion here because it was harder to cram extra info on the slide with the figures. So some of the slides will have extra info in the notes, so make sure to look for that in this PowerPoint if you're using it to study. So functions of the skin. So why are we looking at the integumentary system? What does it do for the body? Why should we care? Our skin provides protection for multiple things. It's protecting us from the UV radiation that comes from the sun, and even can utilize our UV rays to help us with things like vitamin D production. We produce some antimicrobial chemicals to help kill off some of the bad bacteria. We do have good bacteria that live on our skin that we have co-evolved with that help us with that as well. And it protects us from trauma, from friction, right? The outermost layer of our skin is 20 to 30 layers of that stratified squamous tissue that is basically just full of keratin, that keratin protein. And so that is biologically inexpensive, and it can just rub off and protect all the more important functional cells and tissues that we have underlying it. We are going to look at wound healing and the process of wound healing in our skin. Again, it can't continue protecting us if it is damaged and doesn't get regenerated. We'll look at how our skin plays a role in producing vitamin D. So you can get vitamin D through your diet, you can get dietary cholecalciferol, but we also produce it in response to UV rays. We do have sensation from things like pressure, temperature, pain through our skin, and we'll talk about what cells and where the nerves are that correlate with that. Skin plays a role in thermoregulation through sweat and vasodilation, as well as through the hypodermis with fat, providing that little bit of insulation. And lastly, plays a role in some of our homeostasis, so preventing water loss and secreting sodium chloride, secreting salts to help with the concentration of those ions within our blood. So yeah, let's look at how it does that and what we should be paying attention to. So as I mentioned just a bit ago, the actual skin is composed of what we call the cutaneous membrane. So we have our epidermis, which is all epithelial cells. We have a couple different types within the epidermis of our skin. And then we have the dermis, which is underlying those epithelial cells with two different layers of two different connective tissue types. And then under the dermis or deep to the dermis is the hypodermis, which is mainly adipose tissue with some of our arteries and veins traveling through it. Cool. So we'll also be looking at the accessory structures and what role they play in the functions of our integumentary system. So we'll take a closer look at hair follicles and hair, the various types of glands that we have present in our integumentary system, and then a little bit about the structure of nails as well. All right. Let's just keep going. Cool. So first, looking at our epidermis, right? This outermost layer of our skin that is going to be interacting with the environment. The epidermis is composed of 5 different layers or strata, right? The same way that we refer to the layers of different tissue types as being simple or stratified, right? We have various layers in this epithelial tissue. So these are the epidermal strata. And there are 5 different layers. So that's going to be the stratum corneum, being the outermost, stratum lucidum, which only appears in our thick skin, which is the skin on the palms of our hands and the bottoms of our feet. We then have the stratum granulosum, the stratum spinosum, and then at the very base, the stratum basale, which is what is going to have our basement membrane connecting it to the tissue in the dermis. And so when trying to keep these in order, if you could come up with a mnemonic for this acronym CLGSB, I think in the crash course anatomy and physiology video, he uses "come, let's get sunburned." So if that sticks for you, awesome. But again, CLGSB. And so a few of the important parts of each of these layers I've tried to highlight down here in the notes. So essentially, at the very base in stratum basale, we have a majority of these stem cells that are going to be producing the keratinocytes, which will end up producing a majority of the keratin. And eventually those keratinocytes will destroy their own organelles and remove all the cytoplasm and become completely replaced with just keratin fiber in those cells, allowing them to get really squished into that stratified squamous shape that we introduced in the last chapter. So as we work up, these cells are basically going to get flatter and lose more of their function. And so the basal strata here is there to act as those stem cells producing more keratinocytes. That's also where we have our nervous cells, these Merkel cells, which each have a particular nerve or have their own individual nerve that they associate with. And that's how we sense the things through our skin. And so as we move up, again, those keratinocytes will start to produce more and more fibers. And so these next 2 layers, the stratum spinosum and stratum granulosum, are really there to provide some flexibility and stability to the skin overall through that fibrous network. And then as we make it up to the stratum corneum, again, the entire internal portion of the cell is essentially being replaced with just keratin. Will be all protein fiber in there. The nucleus, the Golgi apparatus, the endoplasmic reticulum, they all get destroyed and unraveled, and all of the cytoplasm is replaced by just these keratin fibers. So it's just protein really in that top 20 to 30 layers of stratified squamous tissue that we have making up that outermost layer, the stratum corneum. And so again, in between, if we're looking at thick skin specifically, so just in thick skin, which again is the palm of our hands, the bottoms of our feet, we do have a fifth strata, that stratum lucidum, which is basically see-through. It appears as very glassy and shiny. And beyond that, it really is just there to reduce friction between these other two layers, the corneum and the granulosum, since that is showing up in places where we have a majority of our friction, right? The hands and the feet, which are going to be interacting with our environment on a regular basis or more often than the rest of our skin. That lucidum just provides some protection from that friction so that any friction going on in the corneum isn't also destroying or stressing the granulosum. So besides having that extra layer in thick skin, we also only see these fingerprints or the external expression of these dermal papillae, which we'll look at closer in a second. And that's what gives us our fingerprint. So we have papillae in that underlying connective tissue under the epithelium. And papillae is referring to like ridges, essentially. So let me pull up this image. Okay, so you can see in the papillary layer of our dermis, we have all these ridges. In our thick skin, those ridges actually get expressed through the epithelium to make that fingerprint. And I was trying to look into why this only shows up in thick skin, since we do have these same papillae, we do have these ridges in the dermis of all of our skin. And the only real difference that we can tell between thick and thin skin is that fifth layer, the stratum lucidum. And the only reasonable answer that I was able to find is due to just genetic expression. So amount, how much of the gene is being expressed, or maybe even a different gene being expressed that leads to this physical expression of those papillae. But yeah, that's essentially what makes our fingerprint. It's just these papillae extending all the way through the epithelium, or pushing a boundary all the way through the epithelium. Okay. So that was a majority of the content on our epidermis. And now looking at the dermis, which again is underlying those 5 layers or 4 layers for a majority of our skin. These are 2 different types of connective tissue. So our dermis is split into the papillary layer and the reticular layer. The papillary layer is mainly areolar tissue. And in this layer, really all that I'm asking you to be aware of or to look for is that the veins, capillaries, and nerves that are going to serve our epidermis travel through this papillary or would be present in the papillary layer. Right? So remember that epithelial tissue is avascular. So none of our blood vessels are actually traveling through epithelial tissue. So they are getting all their nutrients and excreting any of their waste through diffusion or through transport with proteins through their membrane. They do not have direct access to nerves or blood vessels. The papillary layer is also what forms those papillae, those ridges, right, which is what makes fingerprints and footprints. So that's areolar tissue, which is a loose connective tissue. The second portion of our dermis is the reticular layer, which has dense, irregular connective tissue. So they're both 2 different types of connective tissue. Even our hypodermis, which we're about to talk about in a second, is a different type of connective tissue, right? It's that adipose tissue, fat cells. And so they're just helping with that connection between our skin, our epidermis, and the rest of our body, and protecting the rest of our body from any friction and damage that may be happening to our skin.
So you said the papillary layer was loose connective?
Yes, or
Thanks.
yes, areolar tissue, loose connective tissue, which was one of the images on your lecture exam. The second tissue example was areolar tissue, and I also was accepting loose connective tissue. And if you were in that range, you got partial credit as well. And then yes, reticular being dense. So we also have 2 different plexi, the subpapillary plexus and the cutaneous plexus, just referring to the branches of the arteries and veins that serve different areas of our skin. So the cutaneous plexus are the branches that supply the hypodermis and the reticular layer. And then our subpapillary plexus supplies the papillary layer and our epidermis. And so again, these plexi or a single plexus is referring to the branching system of our blood vessels. Okay. It's just a closer look at the reticular layer. Something of note, UV radiation, so excessive exposure to the sun will lead to excessive exposure to UV radiation, which can damage multiple parts of our cells, including the DNA. So it can lead to mutations, which would lead to the potential of things like skin cancer. But UV radiation can permanently damage the fibers that are building up the dense connective tissue in that reticular layer. Right, so similar to how we only have a set number of neurons once we've reached full maturation and have generated our maximum number of neurons, from then out, any damage to those neurons will not be repaired. They may be replaced, or a different network may come into play to try to overcome that damage, but it is permanent. You cannot replenish your neurons. You cannot replenish the fibers in your dermis when they are exposed to excessive UV radiation. Something that I find really cool about our skin is that the collagen fibers in that reticular layer bundle themselves into these parallel networks that we call tension lines. And so this is a major application for any medical field as far as making cuts yourselves or treating cuts. So any cut or damage that follows the same line is parallel to these bundles of collagen fibers should heal quite quickly and should have minimal scarring to it, because those collagen fibers are still intact. So the dermis is still intact. And so the repair process, getting all of the nutrients and building new cells, is going to happen much more quickly for a cut that is following that path. A perpendicular cut, something that would cross one of these lines at a 90-degree angle or so, is going to completely unravel that bundle of collagen fibers that was damaged. And so that even causes the skin to pull back even further, because those fibers that were being stretched have now been released, like if you cut a rubber band and both sides of that rubber band stretch to wherever they're being held. So if a cut happens perpendicular to these collagen fibers, that's going to cause the same kind of stretching response that you would see in that rubber band, which can open that cut even further. It takes longer to heal and is much more likely to form scar tissue with that. So I think that's cool. Hopefully some of you guys do too. Okay, and then our deepest layer of our integumentary system is the hypodermis, or the subcutaneous layer. Again, those first three layers are considered the cutaneous membrane. So the subcutaneous layer is below or deep to that cutaneous membrane. And this is mainly just adipocytes, fat tissue. Okay? So storage of fat cells. Their adipocytes are basically completely full of a vacuole, a modified vesicle, a large modified vesicle that is just holding fat. And so that provides a few different functions. The first would be insulation, so preventing the loss of heat or preventing excessive heat getting in, the same way that the insulation in your walls or the drywall is preventing the transfer of temperature, but it's also going to keep in whatever temperature got in there. Okay? So it can help reduce the change in temperature from the environment, but it's also going to help with retaining whatever temperature is internal. And another cool fun fact here, the subcutaneous fat that makes up this subcutaneous layer it makes up 80% of our body fat, or 80% of the fat in our body is making up that subcutaneous layer. And again, that's obviously an average and generalization, depending on how much fat tissue is in your body, and it gets stored in particular areas of the body depending on your biological sex as well. But on average, around 80% of the total fat in your body is found just deep to your skin, so all over your body. Right. How's the pacing so far? Are there any questions, concerns, or things to go back over? Right. So moving along. So another cell that I forgot to mention as a part of that basal layer. So um, we talked about the stem cells that are there creating more of those keratinocytes. And we talked about the Merkel cells, which are there to sense. But we also have our melanocytes, which are melanin producing cells in that basal layer. Melanocytes produce a pigment called melanin. And melanin is what gives the darker brownish and even yellowish coloration that we associate with things like skin color, hair color, eye color. And there's two different types of melanin. We have eumelanin, which produces a brown or black coloration. We have pheomelanin, which produces more of a red or yellow coloration. And we haven't talked much about light in this class yet, but hopefully you have some background context on how light works and how color works from other courses. But essentially, our eyes, we don't see objects. Right? If you completely turn off all of the lights or don't have any incoming light into an area, there's no way for us to perceive what is surrounding us with our vision. Because we don't just see objects, what we are perceiving as what we're seeing is actually the reflection of light off of those objects. And so the color that we associate with something like eumelanin and pheomelanin is actually the combination of light waves that are bouncing off of that pigment. So pigments can absorb certain wavelengths of light. The ones that are not absorbed bounce off, and we interpret those through our eyesight as a particular color. Right? So pheomelanin cannot absorb wavelengths of light that produce the color red and yellow, or that we translate as red and yellow. And eumelanin either absorbs all light or absorbs most light, and that's why we see it as brown or black. And that melanin evolutionarily is there to protect against UV rays from the sun. Right? Those UV radiation rays can damage the fibers of our dense connective tissue, and they could also damage just the individual elements in our cells in general, even causing mutation, which could potentially lead to skin cancer. So having more melanin as this pigment is absorbing certain wavelengths of light and thus preventing that light from transmitting through it and getting to those more important protective parts of our bodies. And so that's why people from locations that are closer to the equator and thus are exposed to higher amounts of UV radiation over time evolve to express more of their melanin or produce more melanin to provide more of that protection from those UV rays. And the farther you are from the equator, the less melanin your body would want because you want to increase your ability to have access to that UV radiation, which is an important part of your vitamin D synthesis in your body, which we're going to talk about in a couple slides here. And so increased darkness or more melanin production does not come from having more melanocytes. On average, you have the same number of melanocytes regardless of skin color, but it's more about how active those melanocytes are, so how much melanin they're producing, and how large they are. So you can see that they kind of extend through this basal layer. And so those extensions would become longer or would be longer for producing a skin type that is darker, producing more and more melanin. And I had the opportunity to learn a bit more about melanin production for a course I was taking in graduate school, and thought it was cool to learn that these melanocytes originally come from stem cells that are basically following the line of our spinal cord, and then they spread from there to reach their locations where they will become their mature melanocytes and produce their melanin. And so that's less obvious in humans, but if you look at like dog breeds or dogs that have different colorations and spots and things throughout their body, right? Dogs tend to have darker coloration along the back, lighter coloration along the belly, and darker coloration at their paws, because those are where those groups of stem cells for the melanocytes are actually located. So I thought that was cool, especially because my dog has a whole bunch of colors, and he is much darker towards his feet, towards his paws, and much darker along his spine and like the crown of his skull, right? And lighter throughout the rest, anyways. So one of the diseases or conditions brought up in the text to discuss related to melanin production is vitiligo. and vitiligo is an autoimmune disorder. So this abnormal production of melanin or abnormal coloration of the skin is because the immune cells are recognizing melanocytes as foreign. So thinking that they are invaders like bacteria or viruses. And so they are attacking those melanocytes and destroying them in particular areas. It's not ubiquitous. It doesn't go across the entire body, or in the cases that it does, that's where we get like albinism. I don't know if that's correct. But for vitiligo, it is just destroying in particular areas, and thus you don't get any melanin production in that area, causing those splotches of skin that you see in that image here. Okay. And so now to the reason why you wouldn't want too much melanin if you're not living close to the equator or living somewhere where you have excessive exposure to UV radiation. That UV radiation can be utilized by some of our skin cells to produce basically the inactive form of the precursor of vitamin D. And this is the same chemical that we are getting through our through food. So our dietary vitamin D is the same as this precursor that our skin produces in response to UV radiation, called cholecalciferol. So that is an inactive form of vitamin D. That cholecalciferol then needs to travel to the liver, which will change it into a different chemical, and then from the liver to the kidneys, and the kidneys will activate that vitamin D as calcitriol. So the active form of vitamin D is calcitriol. And calcitriol is really, really important for our ability to absorb calcium and phosphate or phosphorus from our diets. So without vitamin D, without this chemical, we do still have membrane proteins in our small intestinal cells that can bind with and transport calcium and phosphate. But more calcitriol leads to greater genetic expression of the gene that makes those channel proteins and makes those transport proteins, and so stimulates more absorption of those minerals. And those minerals are really, really, really important for not just signal transduction and activating our muscles, or for creating ATP, right, with phosphate. But that's also what is making up a majority of the mass of our bones, which is the whole structural network of our body. So that vitamin D exposure is really important. And at a certain longitudinal point, I don't remember the exact degree of longitude, for half of the year or so, our planet is on its axis at such an angle. So the winter here in Seattle, right, we are above the longitudinal point that I'm referring to. So during the winter in Seattle here, we are tilted so far from the sun that its UV rays are being refracted to the point that they do not stimulate this production of vitamin D. They cannot transmit through our skin far enough to reach the cells that are producing this compound. And so even if we have a completely sunny day here in Seattle in the middle of January, and you spend all day outside in the sun, you are not stimulating the production of vitamin D. They cannot penetrate deep enough into your skin to do that, which was a new concept for me moving here from Florida. We get intense sunlight throughout the entire year, so any sunlight would be able to stimulate this. But so being aware that we cannot produce this with sunlight during the winter is really important for your management of your nutrients, right? So make sure you're getting some vitamin D supplements into your diet during this time of year, or using things like a happy light. There's other products similar to that, which basically expose you to UV radiation at an intense enough or at an intensity high enough to produce vitamin D. Yeah, interesting stuff there. And so another disorder mentioned by the text is rickets, which is a disorder that comes from insufficient vitamin D production or insufficient vitamin D in the diet. Essentially, this bending that you see of the legs is because the bones are weak. They're not dense. They are lacking calcium and phosphorus to make those crystals that make up a majority of the mass of the bone. And so they're less structurally stable and can start forming those bends that we associate with rickets. Okay, so I'll do another check-in here. Are there any questions about that content or things you'd like me to go deeper into? Okay. So let's look at hair and nails. So hair and nails, you could consider essentially modified versions of the epidermis. Okay? They are mainly going to be flattened keratinocytes with slightly different structure and density that give them their different form as to what we see, right? So even though that nail looks very different and feels very different than the rest of your skin, it's basically made of the same stuff, and same goes for your hair. And so where was that image? Or I have another one. Let's go down here. So your hair stems from the hair follicle, and the follicle is literally an extension of the epidermis down into the dermis all the way to the subcutaneous layer in some cases. So like with how microvilli are using actin filaments to push the plasma membrane to extend outwards, this is basically the opposite. We have extensions going inwards of our epidermis that will form the outer layer of the hair here, or our hair follicle. And so the follicle itself has access to vascular tissue and nerves to supply it. And then we kind of have a circular version of the layers that we were looking at with the epidermis surrounding the actual hair forming in this follicle. So there's lots of different structures to identify. They're colored here, and you can see an electron microscope image of a stained version here. Don't want to spend time just kind of reading the same info, but essentially we have layers that have softer keratin or less dense keratin. We have layers that have harder or more dense keratin, which provides the balance between stiffness and flexibility for those hairs. And we sense a lot of things through the movement of our hair because of the nerves that are connected there at the root hair plexus. So beyond that, I do want to talk about glands. So we have multiple different gland types found in our integumentary system, found in our skin. Okay? Some glands secrete their molecules, secrete their secretions directly onto our hair. And then some glands secrete their secretions directly onto the skin. And so our main categories of glands to be aware of in our skin are sebaceous glands, which are secreting oil or sebum. And we have apocrine and eccrine glands. Eccrine may also be referred to as merocrine. They're referring to the same thing. But apocrine and eccrine glands are our sweat glands. So sebaceous glands secrete oil, which is we call sebum, and then apocrine and eccrine glands secrete sweat. The difference between these two is where they're located in the body or where they secrete to. So starting with the sebaceous glands, they are secreting that oily sebum, which helps lubricate our skin and helps with that protection of homeostasis, keeping our skin cells hydrated, preventing loss of water from our body. And there are no sebaceous glands in our thick skin. So we do not secrete the same oily substance into the palms of our hands or the bottoms of our feet. What you're seeing left behind is any oils that you got from your skin or hair by touching it, following those ridges of your fingerprint. And then apocrine and eccrine glands, again, these are producing sweat. Apocrine glands are only in particular locations, and they only start expressing their secretions after puberty. It's a part of what gets activated through our maturation, through the puberty process. And these apocrine glands secrete directly onto the hair that we have in our armpits and our anogenital region. So it's trying to memorize these: A for armpits and anogenital region. These apocrine glands are only found there, and they secrete their sweat directly onto our hair. And this sweat is very protein rich. So rather than being just water with salt in it, like what is making up the majority of the sweat from our eccrine glands, this has a lot of protein in it. And that protein is going to be consumed or metabolized by the bacteria that we have on our skin in those areas. And that's what produces the odor that we associate as body odor, or BO. Okay? The sweat itself may have more of a sweet smell to it. And then the metabolism from the bacteria is what produces the kind of gaseous waste products that we smell or associate with that smell. And evolutionarily, this is likely a signal to others that the individual has reached maturation, that they have finished puberty, and they are now of reproducing age, right? So acting as pheromones in that way. And we do have a few different examples of modified apocrine glands that have their own name and slightly different secretions. So this includes the ceruminous glands of our ear, which produce earwax instead of that milky protein-rich sweat. It's a modified sweat. We have these ciliary glands of the eyelids, not eyelids, eyelids. And then mammary glands, which secrete milk. These are all different types of apocrine glands. And then again, these eccrine glands, which may also be referred to as merocrine, they're the same thing. Those secrete directly onto the surface of the skin. So apocrine is onto the hair, eccrine is directly onto the surface of skin, and that is more of that watery, salty sweat. So A for armpits and anogenital region, E for everywhere, to try to help with memorizing the difference between these two types. So we have some images showing the breakdown of these things. There is a difference in how these cells secrete their secretions as well, whether it's rupturing the cell itself, basically destroying it, and all the secretions that were inside of it get released, or if we are diffusing or transporting those secretions out of the cell. You're not going to be assessed on those different secretion types. That is kind of exhibited in some of these images here. And then our nails, again, similar to our hair, it's mainly just these condensed keratin fibers to give it slightly different structure. And so we do have our own names for different structures of our nail. Basically, all these folds that you see on the exterior borders of our nail is folds of our epidermis over it. That's what makes our eponychium here at the most proximal end, and then our lateral nail folds on the side. The free edge and nail body only look different because of the transparency of the keratin at that stage. And so you're really just seeing the skin underlying it, or the epithelium underlying the nail body here. And then the lanula, again, is just a different density or different structure of those keratinocytes to kind of give it that slightly whiter color. All right. So our last concept here of different types of injury and repair in the skin. So the main injury repair response can be separated into 4 different phases. And so we'll look closer at all of these, but immediately it's the inflammatory phase. A couple days later, the migratory and proliferation phases are mainly what is occurring. And then the scarring phase can take a couple weeks to a month. So our inflammatory phase is what responds immediately to damage or injury at the skin. So this is where our platelets are going to be called to move towards this damage and start releasing the fibrous proteins contained within them. And we'll start overproducing the protein fibrin, which we just talked about with Marfan syndrome. And the goal there is to create a clot, right, to prevent excessive blood loss. So adding these fibers to clot that blood and prevent it from just excessively leaving or continuously being transported out of the body through those open vessels. Other immune system cells are also going to release histamine, which is going to cause the vessels to dilate and allow for more blood vessels to rush in. And again, with the clot, that'll prevent those blood vessels from just leaving the body and loss of blood. But the goal there would then be to bring in our white blood cells, our immune system reaction cells in our circulatory system, to start destroying any invaders, destroying any damaged cells, so that there's now room to replace them. So this takes about a day or two, or the inflammatory phase is what is occurring for this first couple days after damage, until we start to form a scab and start to replenish some of the damaged cells. So in our migratory phase, the blood clots that were formed with those fibrous proteins are what form that scab. That's just basically a large blood clot. The fibroblast stem cells are going to start migrating down from the epithelium. Basically, we're going to completely recreate the layers, or attempt to recreate our layers, starting with the basal layer. Right, so we bring our stem cells down. those start to proliferate and start to create more of those proteins, those protein fibers that we need to hold this stuff together. So we're basically flooding this with things like collagen and fibrin. The proliferation phase would then be, as the name suggests, where we have a majority of our cell reproduction occurring and proliferating, trying to repair that damage, fill it up with collagen fibers so that we can push that basal layer back up to the surface of the skin and potentially start to recreate the other layers of that epidermis as well. Okay. Our blood vessels will start to regrow as well and start to rebranch out into these areas so we can continue supplying this new layer of cells that we're creating. And then lastly, we have our scarring phase. Again, this would be really, really minimal for any cuts that are small and follow along these tension lines. Okay? Probably an unnoticeable scar if the cut is small enough. But that scar tissue is basically just dense collagen. Right? We've had those fibroblasts producing a large amount of collagen to try to repair that damage and reconnect our tissues so that we can move the basal cell back up to the epithelium. And so what we're left with is that strong network of collagen fibers. So we don't have the same number or types of cells in here that would be functioning the same way that the undamaged tissue would have. It's mainly going to be collagen. And that collagen can be so dense that it even extends above or superficial to our epithelium, and that's what produces these keloids. Keloids typically aren't an issue or cause any additional damage or any additional problems for the skin. It's simply just an overproduction of that collagen as a way to protect you that ends up with a scar that just extends superficially or kind of bubbles up in this way. Then lastly, we'll talk about burns. So you've probably heard reference to things like 1st degree, 2nd degree, and 3rd degree burns. And what that is describing is how deep the damage from that burn went. So 1st degree burns or erythema are mainly damaging just the epithelium. So not getting into the dermis, not getting into the connective tissue, just hitting the epidermis, all of our epithelial tissue above our basal layer or even to the basal layer. And so since we do have nerve fibers that extend and touch or communicate with that basal layer, this can be really, really painful, right? But should be relatively easy, or at least compared to the 2nd degree and 3rd degree burns, this would be the easiest to repair and recover from. A 2nd degree burn is going to extend into that papillary layer of the dermis. And so again, this would be painful. We do still have nerves communicating with that. Since this is much deeper, we don't have the exact same repair response that we would for our epithelia, and requires a lot more biological currency, right? A lot of energy and molecule input to be able to completely repair that papillary layer of the dermis. And so healing takes a bit longer. And then lastly, 3rd degree burns extend all the way to the subcutaneous layer, to the hypodermis. And so strangely, this would be likely the least painful of the burn types, because you've essentially burned up any of the nerve endings that would have produced that damage response, or have produced that feeling of pain, right? Because the burn has extended all the way to the subcutaneous layer, completely removed your entire portion or damaged your entire portion of your skin here. And so you don't really feel the pain, though this is the most severe because it is the deepest and thus requires the most care to repair. Right? Everything in here has been destroyed or would need to be broken down and then repaired. And so that's likely where you would get a skin graft, so a complete replacement of that tissue from some other stem cell or from another person, right? Rather than relying on your body's ability to replenish that, because it may not be able to. And the last thing to talk about here is just a discussion of this rule of nines. And so anyone who may have more experience in the medical field may be able to tell me how much or how prevalent this actually is, but they do discuss it in the textbook of separating the human body into percentages based on how much of your skin is in those areas. So 9% of your skin is covering the head. There's 9% of your skin covering each layer. Your trunk composes 36%, so 9 times 4. Right? And then each of the lower limbs has 18% of your skin. And that's different for a child, doesn't follow that exact same 9% pattern, but that's where this rule of nines idea comes from, 9%. And the idea with separating this is to address how life-threatening a damage or a burn would be, because a burn that is covering 20% or greater of your skin, or that damages 20% or greater of your skin, would be considered life-threatening, very serious, and needing immediate attention. So that's the idea there. Yeah, does anyone have more context to that as far as if this ever comes up in an actual medical setting?
I used to assist a dermatologist at the UW, and we used it every day.
Oh,
He
cool.
would chart in his notes, like if you were coming in for like psoriasis or a rash, like how much of your body surface was covered by that. And we had like these diagrams for the rule of nines in our office.
Okay, good to know. Thank you for that insight. Okay, so that covers the majority of the chapter and covers the specifics that I want to make sure you are addressing in this chapter. Constance, were you going to say something?
Oh yeah, just about the upper limb, you said 9% on each upper limb. I believe it's 4.5% on each upper limb, and the 9% is for both upper limbs.
Okay, let me double check what was in the textbook, and the textbook could be wrong too. But that's just
And I
what was
think
on that.
the lower limb is 9% each.
Okay. So where I'm putting 9% each, it should be 9% combined and 18% combined.
Yes, correct.
That would be 36 plus another 36, 60, 72, plus another 18, be 90 plus 9. I mean, the math adds up, but like I said, I'll double check the textbook. Yeah, any other insight or comments here or questions to address?
Oh, can we just go back to the proliferation phase real quick?
Absolutely. Did you have a particular question about it or just wanted to see the slide again?
I just missed it, so I was just going back to it.
No problem. Yeah, the text is backing up those same numbers. Let's see if a different source talks about them differently.
What I saw online for the rule of nines was that the front side of the arm is 4.5%, and then the back side of the arm is also 4.5%. So then it equals 9, but so the front is 4.5 because that's kind of what the diagram is showing. But
Cool. Yeah, PubMed backs up that same idea too. So these should be accurate. Hopefully these are accurate. Constance, if you have something else that backs up what you're saying, I'm happy to take a look at that so that we can all get onto the same page with it. But based on what I'm saying, this should be accurate. Thanks, Ben. So again, any questions, any other information you need to see from this? Anything you guys have heard of or seen that you wanted to ask questions about or discuss related to the skin?
so in the proliferation phase, the scab is going away and the scar is starting to form. Is that right?
Yeah, so basically the macrophages are starting to eat up that clotted blood. So the scab is starting to dissipate as we eat up more and more of that clotted blood and destroy it. And then fibroblasts are creating these granules and creating more and more collagen fiber that's going to help push up that basal layer back towards the surface to try to become even with the epidermis again.
Can I see the diagram of the nail again?
These are in your text, so which one did you want? A, B, or C?
This one.
Yes, all the figures here are straight out of the textbook. So you guys have that there, with a bit more description about each of them. I'll have clearer information for you as far as what to focus on here. But for now, any of these structures are fair game. Get one last call for any other info from Skin, and then I will take this opportunity of these last 30 minutes to talk about the exam, since everyone here was here for the exam yesterday. Okay. Cool. So if you missed it when I was talking about it at the very beginning of class, I was able to finish grading all of the exams. So there is a column in the grade book called the Unit 1 Lecture Exam Grade, which just shows the percentage that you scored on this first exam. That will be combined with your percentage from the skeleton lab exam to create your average, which will be your Unit 1 exam grade. So as far as like the class policy where you need to score at least a 40% on any one exam and maintain a 50% average, right? This one score is not necessarily describing where you land there yet. It's going to be the average with your skeleton lab exam to create your one exam grade. And hopefully that's clear enough. So I'm going to pull up my key. And some things I did not stick exactly strictly to based on how I felt about the question, based on the responses I was seeing. And yeah, I mean, I guess that's essentially it. The only time I was flexible with it. And you got partial credit a lot of the time for being close to these answers. Again, I care much more about you showing an understanding of the concept, the relationship, the pattern, than I do about exact specifics. So I tried to give partial credit anywhere that seemed relevant. So is everyone seeing the exam key here? Cool. Oh, I guess I don't think I want to record this portion. So I want to be able to use this stuff later. I want to reduce the ability to share this info. So I am going to stop recording. This will not show up in the recorded version of the lecture, so that we can talk about it.